In our last lecture, we started talking about the structure of a skeletal muscle, and we talked about the fact that a muscle is really a bundle of fibers, and those individual fibers are the actual muscle cells. So when you think of a muscle, you probably think of what we consider to be the muscle belly, the large part of the muscle. So the biceps, the quadriceps, the latissimus dorsi, these are all what we consider to be skeletal muscles. But those muscles are actually made up of smaller bundles of fibers that all work together to cause movement, right? So let's take a look at this diagram here. This is a good breakdown of how a muscle is set up. It is set up in essentially, like I said, bundles called fascicles. So these individual fascicles is a bundle. They are bundles of fibers that work together as a unit. So this would be like a muscle here. So this would probably be one of your quadriceps muscles based on where it's located on the femur. And as you take a look at it, you can see the outside of it is covered in a material called fascia, which we talked about. And that fascia becomes the tendon which attaches the muscle to the bone. And that fascia is what covers the entire muscle and keeps it as one unit, essentially. And then if you take a look, you can see that these muscles are actually broken down into smaller sections. So there's a membrane around the entire muscle called the epimysium, which is the outermost covering. And then those individual bundles of fibers, which are called fascicles, those are going to have another membrane around them called the perimysium. And then finally, the individual muscle fibers, which you can see in this image right down here, where the arrow is, that would be a single muscle cell. So a single muscle cell is bundled together with the other muscle cells in a fascicle, but the individual muscle fiber has a membrane around it called the endomysium. And the endomysium is going to be over what we call the sarcolemma, which is the membrane of this muscle fiber. So you can see here in this image, this is where we left off in the last lecture. This would be an individual muscle fiber, which is made up of a membrane, which is the sarcolemma. It's got multiple nuclei. So muscle fibers have multiple nuclei, and that is because at one point there were individual cells that during development fused into one long cell, so they cannot replicate because they have multiple nuclei. And this yellow material here represents that sarcoplasmic reticulum, which we talked about in the last lecture. And then finally, what makes up a majority of your muscle, and I'll slip to the next slide. This is kind of zoomed in on that image, are these bundles of fibers. And these fibers are protein filaments. They're made up of protein, and there are two types that we're going to really focus on when we talk about a muscle. They're called actin and myosin. And they're called myofibrils. And myofibrils are just long chains of proteins which are made of amino acids, and there's a thicker one, which is the myosin, and a thinner one, which is the actin. And it's the relationship between those two types of proteins which makes a muscle actually function. So when you think of a muscle cell, you should think of a long, thin cell made up of mostly protein fibers that fill it up. So that's what we left off in the last lecture. So let's take a look even farther. So this is just another image here that shows you an individual muscle cell. And you can see this would be a fascicle here, right? So a fascicle is a bundle of muscle fibers. So this would be the perimysium, right? The perimysium on the outside. And then this would be an individual muscle fiber, which is what we consider a muscle cell. Let's talk a little more about what's going on inside this muscle fiber. So this is just another image, and what you could see here is the sarcoplasmic reticulum. So you can see that surrounds these fibers, and we also have the nucleus and the sarcolemma, which is from the last slide. But we have some new content on this slide, so let's take a look at it. The first thing I want to point out to you is there are these little enlarged areas on the sarcoplasmic reticulum. They're called cisternae. And what a cistern is basically just a reservoir. So for example, if you've ever traveled anywhere that has like an island location, like down in the Bahamas, or if you've ever gone to like the Virgin Islands, they don't have plumbing a lot of those places, or at least they don't have a good water supply. I shouldn't say they don't have plumbing. But what they have is on top of the roofs, they have these big giant barrels that catch rain. Those are called cisterns. And a cistern is just a reservoir. So in this case, this is also a reservoir, but it's in a large portion of the sarcoplasmic reticulum. And what it does is it holds on to calcium. And we're going to talk more about that later in great detail. But these little reservoirs of sarcoplasmic reticulum, they're enlarged areas which allow for the accumulation of calcium. And I talked about how important calcium is before, and we're going to get way more into that as we go through. One other thing I want to point out to you are these little things called T-tubules, or transverse tubules. They're little holes on the outside of the muscle. So if you take a look here on the outside of the muscle cell, there's those little holes. And what they are, they're not just holes, they're tubes. And they're tubes that run deep into the muscle fiber. And they're called T-tubules. Here's the thing about a muscle. A muscle without a nerve attached to it is a useless piece of meat. Right? Because in order for a muscle to contract, it needs to be stimulated. And it can get stimulated really two ways. One is through a nerve stimulating it from your nervous system. And the other way is you could get electrocuted, right? If I jam my finger into an electric socket right now, all my muscles are going to contract. And that's because of that electrical impulse. So ideally, we want to control our muscles. And the way we control our muscles is by allowing an electrical impulse from our neurons to travel deep into the muscle. Well, here's the problem. The muscle fiber is thick, actually. So if you take a look here, this would be an example of a muscle fiber. And as you can see, there are muscle fibers deep in this muscle here. So somehow we got to get the impulse from the outer portion of the muscle cell deep into the muscle fiber. So what these T-tubules do is they provide essentially a tunnel for this electrical impulse to go deep into the muscle. So just so to help you understand why these T-tubules are important, if we did not have them, that's a good way to think about it. Let's say we didn't have T-tubules. What would happen is if this muscle fiber got stimulated by a neuron, it would just stimulate the outer fibers. So the outer muscle fibers would get stimulated, these actin and myosin, but the ones in the middle would never get stimulated because they wouldn't have any way to get the impulse there. So the T-tubule really is just allows the impulse to go deep into the muscle fiber so you get a strong contraction. The difference between a weak contraction and a strong contraction is the number of muscle fibers that are stimulated. And we're going to talk more about that as we go through. And finally, the sarcoplasm. And the sarcoplasm is just the cytoplasm. And we call it the sarcoplasm because it's in a muscle, but it's just the cytoplasm. So just to kind of go over what's on this slide here, we have our sarcolemma from before. We have our nucleus, which we identified. We have the sarcoplasmic reticulum, which is what allows ions to travel through the muscle fiber. We have these enlarged portions called the cisternae, which have the important job of accumulating calcium, which will become much more apparent as to why that's important as we go through this. And then we have our T-tubules, which are very important because they allow the impulse to travel deep into the muscle. Okay, we're going to finish up here with a really important concept called a sarcomere. Now here's the thing about a muscle. A muscle is very long. It's a long fiber, a long, thin, essentially tube of these fibrils, these myofibrils, actin and myosin, but they're actually broken into segments called sarcomeres. And a sarcomere is tiny. So one muscle fiber would have hundreds of thousands of sarcomeres, and that's where the muscle fiber actually collapse. So let's take a look at, this would be a sarcomere here. So this would be like an individual muscle fiber, a single myofibril. So inside a muscle cell. So this would be a muscle cell here. This would be one muscle cell, and inside that muscle cell, we have thousands and thousands and thousands of these long, thin fibers, which are made up of actin and myosin. And the actin and myosin are what cause the muscle to contract. And we're going to talk about that a little later on. But we're just talking about the anatomy now. And what you end up with is an area made up of these things called zones. And a sarcomere, an individual sarcomere, is the part of the muscle that contracts. So what makes a muscle contract is they all contract at the same time. So this gets shorter here, this gets shorter here, that gets shorter. So the entire thing kind of collapses on itself. So the best way to think about a muscle is an accordion. If you've ever seen an accordion before, it's an instrument that you can kind of open and close. And when it opens, it pulls air in, and when it closes, it pushes air out. And it has these little ridges that allows it to open and close. That is exactly how a sarcomere works. This sarcomere would be relaxed in this state, and then when it shortens, these little areas here move towards each other, and that makes the muscle contract. So let's take a look at what we got going on here with a little drawing. So I drew this a little bit earlier. So what we have here is this would be a single sarcomere. And a sarcomere is multiple components. So let's take a look at what's going on here. The first thing we have are these little guys, which are actin. So actin fibers are much thinner than myosin. They're thin. They're actually a double braided protein, so it looks almost like DNA. It looks like a double helix in the sense that it wraps around itself, but it's a thin protein. And then myosin is a much thicker protein that has little heads on it, which we're going to talk about later. But the important thing for you to know now is myosin is thick. So that would be our myosin. Now there's a couple structures here I want to point out to you. The first one is called the Z-line. And the Z-line is just a protein which anchors a sarcomere. So a sarcomere is going to be between Z-line and Z-line. So what would be next to this would be just another one of these. So you would see another sarcomere over here and another sarcomere over here. But there's a couple areas. What you're going to notice here is what makes these striated is that there's some difference in the thickness and thinness of these filaments, and what that leads to is a light and dark area on a slide. So what are we looking at here? I'm going to bring back that PowerPoint just to kind of show you an actual. So this would be an actual sarcomere here. So an actual sarcomere on a slide, this would be a Z line here, this would be a Z line here. And the Z lines are the ends of the sarcomeres. And then what you'll notice is there's some light areas on here and there's some dark areas on here. And what makes them light versus dark is the type of protein that's there and whether or not they're overlapping with each other. So let's just go back to my image here and we're going to draw it in. Okay, so the first thing I want you to notice is that there's some areas where they overlap, right? So actin is thin, myosin is thick, and in some areas, like for example, I'm going to bring over these little dotted lines here to show you, there's some areas where they overlap each other, and there's some areas where they don't overlap each other. So I'm going to just kind of drag in these dotted lines. And what they show you is that there are some boundaries, right? So for example, this area here is made up of just actin. So that's going to be a very thin protein, so light will pass through it very easily. This area in the middle is just myosin, so it's thicker, so not as much light's going to pass through because it's a thicker protein. And then finally we have this area here, which is made up of where actin and myosin overlap, and that's going to be extremely dark. So what we're going to have here is we're going to have these bands that get created. So if I go back to my image here, just so you can see, this would be the area that's just actin, this would be the area that's just myosin, and where it's the darkest is where actin and myosin are together. So let's just go over the terminology, and then that'll be enough for this lecture. Okay, let me just bring back my image. All right, so we have, first off, we have what is called the I band. And the I band is going to be the area that is just made up of actin. So it's going to be the lightest color. So this would be our I band here. This would be our I band here. So if I were to color in the I band, which let me see if I can get a nice light color here. Well, we'll just go with that. Okay. And then the A band is going to be where actin and myosin overlap. That's going to be your A band, and that's going to be the thickest band. Now the M zone is going to be, or the M line I should say, is going to be right down the middle. So the M line is just right down the middle, and that anchors these proteins, and that's easy to remember. M line stands for middle, Z line is the end. And then this area in the middle where it's going to be just myosin is what we call the H zone. So this is the H zone. So it's H zone where it's just myosin, A zone where it's actin and myosin, and I band where it is just actin. And that is what we call the sarcomere. And when this muscle actually contracts, what ends up happening is the Z line gets pulled inwards. So what you end up with is this would shorten in this way, this would shorten in this way, and the muscle will get shorter. So I know it's a lot, but let's see how we do with that. So just to finish up here, just the terminology. So we have this would be one single sarcomere here, Z line to Z line. This would be a second sarcomere here. So this would be the Z line here, just as a reference point. So the Z line is the end. The M line is the middle. The I band is going to be on either side of the Z line, and that's just actin. So this would be our I band here, where it's just actin. I band here, I band here. The H zone is on either side of the M line, and that's just myosin. And then the A band is where the actin and myosin overlap. So a sarcomere is essentially a lighter section of the I band and a darker section of the A band with the H zone in the middle. And that is what makes muscles look striated. That's why they look light, dark, light, dark. Each striation is actually a sarcomere. Thank you very much.